Thanks everybody for coming out. As you all know, I'm from past seminars, I'm not the best at giving introductions, so I made a few notes this time. I'd like to welcome Dr. Zenithia Barnett with us today. He's a research fisheries biologist with the U.S. Forest Service Southern Research Station. She has a bachelor's from North Carolina Ag and Tech, master's from University of Florida, PhD from University of Mississippi. She said she's currently a fish research biologist with Southern Research Station, based out of Oxford, right? But will soon be based right down the hall here at Clemson. We're happy to have her, and I won't take up any more of your time. Ready to hear what you have to say. Thanks.
Thank you, Brandon. And today I will be chatting to you a little about crayfish conservation through land and water management decisions. And as Brandon said, I am a research fisheries biologist with the USDA Forest Service, and I will be relocating here to Clemson soon. And I'm from South Carolina, so I've been trying to get back home close to the family. I grew up in the coastal area, so my family's from Charleston, the Charleston area, but I grew up in Estill, South Carolina, and Hampton County, so a lot of roots in South Carolina. So a big push to come home, do some research at home, learn about the streams and the fish and crayfish where I grew up playing. So it's kind of a joy to be able to relocate and come back this way. And so I'm going to start off talking about the imperilment of our freshwater systems in the United States. And so this map shows the amount of imperilment throughout the United States, with those dark red colors showing very high imperiled systems. And so as you can see here, out west in California and the southeast are where we really see this high imperilment. And so our freshwater systems here have very high imperilment, which is where we find basically we're losing species. Species may be only found in certain drainages or in small areas, but there's a lot of loss of species within these areas. And so our threats to our freshwater system is a lot higher than we find in our terrestrial or our marine ecosystems. And that's because a lot of different reasons. But basically everything we do affects our freshwater system because of watersheds. So if you do anything in your backyard, it slowly makes its way when it rains to a ditch, which may go into a stream, and all of those organisms are impacted. So things like urban development, pollution, logging, damming, and water management and agriculture all impacts our freshwater system. So we have higher threats to these organisms than we find in our marine and terrestrial ecosystems. And so this map shows crayfish diversity. So that's my main study organism are crayfishes. And crawfish, as we often say in the South, they're the same thing. And the dark blue in this map shows where we see the highest amount of crayfish diversity. So as you can see, that dark blue really only resides in the southeastern United States. So if you think about that imperilment map with that dark red being in the southeast and this dark blue for crayfishes being in the southeast, just as that imperilment map shows, we have a lot of imperiled crayfish species in the southeastern United States. So there are more than 300 crayfish species in the United States, and 80% of those are in the southeast. And just to kind of give you an idea of how many species are in the southeast, Alabama alone has 100 different crayfish species. So 1/3 of all the crayfish diversity in the United States can be found in one state. And so the southeast is the crayfish hotspot, as I like to say. But thinking about that imperilment map, over 51%, only 51% of crayfishes, so over 51% of crayfish species are of some type of conservation concern. So from vulnerable to presumed extinct. So we have 300 species in the United States, over 150 of them show some type of conservation concern. And crayfish is the second highest taxa that of conservation concern in the United States. So we do have a lot of different, we're losing a lot of different crayfish populations, crayfish species, and we need to find ways to manage so that we can protect these species. And one reason I think that we're losing a lot of species is because crayfish live in all types of habitats. We have from our streams, our lotic species, which we can find in our streams, in our rivers, our lentic species that can be found in our lakes, in our vernal pools, in ditches. And then we have our terrestrial or subterranean species that groundwater is their main water source. So they are found in agriculture fields, in prairies, in our riparian zones, but they don't need a surface water source to survive. But basically, normally I tell people when we're in the Southeast, no matter where you're standing, you're likely probably close to a crayfish. So you can be in any type of water source and not near a water source, and there could be a crayfish present. So anything we're doing on our land, any changes we're making could potentially be impacting these populations. So there's a big importance of our land management to these populations, and forested systems are extremely linked to our freshwater ecosystems. And if we first think about our surface waters, so our rivers, our lakes, those waters, they exchange flows with these riparian zones and these forested systems. So when the streams flood, they exchange nutrients. When leaves fall, when wood falls in the stream, it's creating habitats and food sources for these organisms. And it is very important for what lives in the stream and plays a lot in the diversity we see in the streams. These forested systems, by protecting the substrate and holding substrate in place, we have larger rocks and things in these systems, not a lot of runoff, and we find more spaces and things for crayfish to live under, to hide, to use as protection from predators. And in our flooded systems or in our forested systems, we also have vernal pool habitats that are very important to burrowing crayfishes, and they protect groundwater sources. So all of these things are intricately linked to our crayfish populations. And so someone may think, well, you know, these systems are important to crayfish, but how are crayfish important to these systems? Why do we care if it's 300 species versus just 150 species? What changes, what might we see in these systems if we lose crayfish? So I think of crayfish as the backbone of the system. So almost over 200 taxa eat crayfish. So if you think about the bass and catfish that we like to eat, studies have shown that over 40% of their diets can be of crayfishes. So if you lose crayfishes, all of these higher taxa will be impacted as well. And you have from coons to otters to birds to pigs, all of these animals eat crayfish. So they're a very, very important food source in these systems. They're also omnivores. So they eat both plant and animals, and they play a big part in managing the amount of vegetation that are in these systems. So they eat a lot of vegetation, they eat a lot of algae, and they play a big part in how much vegetation are in these systems. And they also are ecosystem engineers. And so their burrowing capabilities, these, what you're seeing here are crayfish chimneys. And so these are balls that they build up on top of their burrows that are down under the ground. And so with them digging these burrows and bringing sediment and out of the ground, they're making nutrients available to organisms that wouldn't naturally be available. They're creating burrows that other organisms use, from salamanders to, we have a crawfish frog that only uses crawfish burrows. So if you lose these burrows in a system, you can lose this species of frog. But their sediment manipulation really impacts these systems and the animals that live there. And then there are also bioindicators. So there are some species, not all, but some species that are sensitive to pollution, that if you find them in your system, that it's an indicator that you have a healthy ecosystem. So I just talked about how over 51% of species are of some type of conservation concern. But if you look at the Fish and Wildlife Service list of endangered species or threatened species, you'll only find 6 crayfish species on there. So if you look at it, it looks like, oh, these crayfishes are actually doing pretty good. You know, there's only 6 listed out of all of them in the United States. But that's definitely not the case. So there are 4 species proposed for listing. So there are 4 species that they've gone through, done assessments and say, yes, they do need to be on the endangered species list. So hopefully soon we'll have 10 species on the list. But there are over 90 species that are under review. So there are 90 species that we that have been petitioned, that there are currently work going on to figure out where they're located. Do we think they're endangered or a conservation concern, but we haven't really looked the right places? Are these populations truly declining? And if they are, what are impacting these populations and how can we try to protect them? So there are over 90 species that are currently being worked on to potentially be federally listed. And so one of the big reasons that, or one of the things that I've studied that has a big impact on these populations is habitat fragmentation. And that is from our terrestrial to our aquatic systems, and all animals really face habitat fragmentation. And this is when you take a big continuous piece of habitat and split it into smaller sections. And so in our terrestrial system, this can be done by roads, by agriculture fields, through urbanization. And in our aquatic systems, you find it done through impoundments, culverts, weirs, rail lines, but things that basically stop the organism from freely using the entire area that it used to have available. And so I'm going to talk a little about fragmentation within our terrestrial system. And so in the South Carolina Piedmont region, near the Sumter National Forest, we find the Distocamberus genus. And this genus is, a number of the species within this genus is up for listing. And so we're doing a lot of assessments now to see if the species or some of these species should be listed as threatened and endangered on the Fish and Wildlife Service list. And these species live in open canopy terrestrial habitats, historically in Piedmont prairie habitats. And if you think about South Carolina, I'm sure you do not think about prairies, but there used to be prairie habitats in South Carolina that these organisms use. And present day, they're found along in roadside ditches, in utility right-of-ways, and in agriculture fields, and then food plots and open areas. And so the National Forest has been managing land to create habitats for these species through more burning of their habitats to create open spaces. They've been found living within dove fields, so fields that have been managed for have open spaces, and in food plots. Management that has been done to create open areas for food plots for other organisms is where we currently find these crayfish species. And so for this study, we were focused on Distocamberus crocori, and we sampled within the Long Cane, on and off the Long Cane Ranger District of the Sumter National Forest. We sampled vernal pools, ditches, and those key open habitats for burrows that I just discussed. And then we did a genetic analysis to look at the population genetic differences within the species. And so we collected tissue from legs and used the mitochondrial CO1 gene as our marker. So this map shows the genetic structure of the Distocamberus species. Each dot shows locations where we collected the species. And so we sampled more than where the dots are, but the dots are where we did find the species. The color of each dot represents a genetic population. So if the dots have the same color, that means their DNA sequences were similar, which means that the crayfish in those different populations have been mating and exchanging genetic information. The background of the map is of the physiographic region or the ecoregion. So as you can see, most of the crayfish we're only finding on the Carolina slate belt, which is that lighter gray color. And so the first thing that we saw is that basically the species is only found on the Carolina slate belt. So if we're going to manage for the species, we need to target the Carolina slate belt. And so we have a northern Carolina slate belt and a southern Carolina slate belt. And this is a burrowing species, so we figure soil type and groundwater level will likely be a big factor in where we're finding it. And the soil types here were granite in the northern portion, and then Carolina terrain in the southern portion. And so basically these areas though have large, flat, erosion resistant areas, and they have a perched water table. And so the water table here is a lot higher than in the surrounding areas. So if crayfish are burrowing down, going to the water table, they don't have to burrow down as far. And so they like these this type habitat. And so if we look at the genetic structure though, we see that basically the southern habitat has this basic continuous one genetic population. So crayfish look to be moving all throughout the southern region. And in the northern region, we see 3 different populations. And what was most interesting to me is the black and orange population that were basically right on top of each other, but 2 different populations. And they were actually collected along the same ditch, but the genetic structures were completely different. And so, you know, questions came to, you know, why do we see this continuous population in the southern region, but this disjoint population in the northern region? What's happening there? And so we went on to look at land use differences. And so the background of this map has changed. The dots are still the same in the same spots, but now we're looking at land use differences. So the greens that we see are our forests, the pinks and reds are developed areas, and our yellows are agriculture. And so what we see is that that red is all throughout our forest. So that red is the Sumter National Forest. And so it looks as if the forest, that forests facilitate gene flow, and that the crayfish are easily able to move throughout these forested systems. And we weren't sure if that was actually going to be the case because we really don't find them burrowing in the forest. So we didn't know if forests would actually, they would or wouldn't be able to move throughout the forested systems, but it looks as if they can really move and make, you know, throughout these forested systems. But in the, where you see the black and what used to be orange, but looks kind of yellow now. At the top here, the northern region, most of that region is agriculture. And so it looks as if agricultural lands resist gene flow. And so it could be hypotheses are that, you know, could be herbicides or pesticides that they could be using, or ditch management, because most of these crayfish were collected in the northern region, were collected in ditches. So if they are digging up ditches during certain times of year for management, or if there are different pipes in these ditches that crayfish can't pass through, but something with this ditch management is affecting these crayfish, and we need to consider ditch management when managing for them. Okay, so I'm gonna jump from fragmentation within our terrestrial systems to fragmentation within our aquatic systems. And focus on impoundments and our dam population and dams within these flowing systems. So there are over 87,000 dams that are greater than 3 meters tall within the United States, and over 2 million that's less than 3 meters tall. And then Alabama has greater than 10,000 dams. And so this study and focus is going to be in Alabama. So you see the Alabama map there with all the blue dots representing all the dams around Alabama. And as I told you, Alabama has the highest crayfish diversity in the United States. So we have crayfishes all throughout the state, but a ton of dams and things that could be fragmenting those habitats. So impoundments were all built for very, very great reasons: to control flow, to prevent flooding, to provide energy, and transport goods. But we clearly weren't thinking about what it would do to our stream systems and if the organisms in these streams would be as happy about these impoundments as people were. And so what we see is that they impacted the connectivity. So organisms are normally stuck on one side of the stream or another, so they stuck in a stream section. They reduce connectivity to our floodplain, so if it prevents flooding, then you no longer have that connection to your riparian zone. And then it flow regime and temperature regimes also change. And so most of our stream organisms, our fishes, our mussels, our crayfish, the times that they mate, the times that they release young are all connected to flow and temperature. And so if that changes, then you really will see a change in what organisms are living in those areas. And so some of the biological impacts that have been shown through various studies are changes in spawning, larval survival, and growth patterns. So studying this study was one of my PhD studies, and these actually, the publications that have come from that PhD work are the only publications on how large impoundments impact crayfishes. And so if you think about all the impoundments we have in the world, how long they've been around, all the research that's been done, it's kind of mind-boggling to me that nobody's assessed how dams impact crayfishes. So if you're trying to think of a new study and what we need work on, this is definitely a path to go down, because there's lots and lots of work to be done to truly understand how these populations have changed and way that we can manage, because they are removing dams, but a lot of them won't be removed. So ways that we can manage to promote and the health of these populations. So for this study, we looked at 3 dams within Alabama, 2 in the Bear Creek drainage, and then 1 in the Cahaba River drainage, and then we also had unimpounded drainage for comparison. And so one thing with working with crayfish is there aren't a lot of reference studies or reference information of the streams before historically. So we didn't have reference data to be able to look at what the stream originally, how the populations were originally in these streams, but we compared that to the unimpounded streams as our reference. And so for this presentation, I'm going to talk about one crayfish species, the Vaxonius validus, in the Bear Creek drainage, and look at the genetic structure using the same mitochondrial DNA gene that I used with the distal Canberra species to assess how connected these populations were within the stream system. And so the dots here are pretty similar to what I previously discussed, where each dot represents a site where we collected the species, and then the color of the dots represent one single genetic population. And so for Rock Creek, which was our unimpounded stream, we saw that there was one continuous population within these streams. So crayfish seem pretty happy within the stream system. They're moving from up to downstream without any problems and mating with each other. So we saw one continuous genetic population here. But when we look at Cedar Creek, which is our impounded stream, one of our impounded streams, we see 5 different populations in the stream system. And so these streams actually flow north. And so our downstream population is this black population. And then we had 4 different populations upstream. And while there are lots of populations within the stream, what I want to point out here is that there are no overlaps. So none of the populations shown downstream are found upstream, and none of the populations found upstream are also found downstream. So there looks to be that there's no movement between throughout the impounded system. Crayfish are really stuck to one stream section or the other. When we look at our second impounded stream, doesn't really tell the same story. So we have one main population, which is this green population that we found both up and downstream. Then we have a black population found downstream and a yellow population found upstream. And so while we do have, it does look like crayfish can move throughout these systems. They are, we do see these, dish jump populations popping up, so there could be some type of fragmentation occurring that, that just hasn't, really shown yet. And so these dams were built in the 70s, and so for a genetic timeline, it really hasn't been there that long. And so it was really surprising that, okay, we were able to detect this fragmentation so quickly with the dams being built in the 70s. But we still left with the question of why do we see this fragmentation definitely occurring in one stream but not the other. And so my hypothesis is it has to do with the size of the impoundment. And so these impoundments, as I said, were both built around the same time, maybe 2 to 3 years difference. But Cedar Creek impoundment is 3 times larger than Little Bear Creek impoundment. And so we have a larger lentic area, a larger lake that these crayfish would have to maneuver through. So instead of a flowing system, they have a totally different system that they would have to maneuver through in order to get from up to downstream. And so basically, the larger the impoundment, the more impacts that is having to these systems. So we were also interested in how did the crayfish assemblage change? So we saw the genetic structure did change in one of our impounded streams, but what about the other crayfish? We were only looking at one there. How does crayfish assemblage change in our impounded and unimpounded streams? And so this is the NMDF plot with each shape representing the assemblage collected at the site. So all the different crayfish species and with their abundances at each site. The circles represent our unimpounded stream, and the triangles and squares represent the impounded stream. The unfilled shapes are sites upstream, and the filled shapes are sites downstream. So the first thing I want to point out here is that we did see differences between the unimpounded and impounded stream assemblage. So there wasn't any overlap in shapes. And so what I should say here that I didn't say, the closer the shapes are together, the more similar the assemblage is. The further they are apart, the more different they are. And so we didn't see any overlap between the assemblage at our impounded and unimpounded streams. So the crayfish species that were collected here, the assemblages were really different. So the impounded streams have changed their assemblages. And then we also saw that there was a difference between sites up and downstream in our unimpounded stream. And so in our unimpounded stream, upstream, Canberra striatus and Faxonius validus were the 2 main crayfish species collected. And so as you shift from up to downstream, the percentage of those crayfishes slowly change, where once you got downstream, Faxonius ericksonianus and Faxonius compressus were the 2 main crayfish species collected. And so there's this shift in assemblage in the unimpounded stream. But in our impounded stream, no matter where we were, no matter what we collected, no matter if we were up or downstream, Faxonius validus and Faxonius ericksonianus were the 2 main crayfish species collected. So it looks as if the impoundment has homogenized that crayfish assemblage within those impounded streams. And I do want to point out our species that we seem to be losing are habitat specialists. So Canberra striatus, which is abundant upstream in the unimpounded, they use our floodplain system. So they're secondary burrowers. So they're burrowing in the riparian zone during parts of the year, and then they're in our stream during other parts of the year. So flooding is really important for this connection, and if you're reducing flooding with the impoundment, then you might lose these type species. And then Faxonius compressus, which is was abundant downstream in our unimpounded stream. It likes small ripples with lots of pebbly habitats, and we didn't see a lot of those habitats in our impounded stream. And so our impounded stream, those habitat specialists weren't doing really well. The more habitat generalists were doing well. And I call these 2 species habitat generalists because we also collected them within the lentic system, so within the lake. So they also able to survive within that lake area. And so they can survive in lots of different habitats, and those that aren't really, the species that specialize, those were what we were using in the system. So what is driving this, these differences? What environmental factors did we see that were driving the changes within our assemblage? And here are kind of the shapes are flipped. So our triangles represent our impounded stream, and that's shown at the top of the NMDS plot. And our squares represent the unimpounded stream. But again, each stream represents a site. Closer they are together, more similar; further away, more different. And then our vectors shown here show the environmental variables and that are the direction that they're driving the assemblage. So what we see here is that there were lots more predators, so fish predators, bass, catfish, and other fish predators that eat crayfishes. There are a lot more predators in our impounded stream, and that seemed to be driving the assemblage. And these impoundments are stocked with fish because there's lots of fishing that happened there. So people are actively putting crayfish predators within these streams, and so that could reduce the what crayfish species and abundance that we're finding there. And then there was a higher minimum discharge, so the water levels in the streams didn't get as low as it would in our unimpounded stream. And then in our impounded stream, there was a higher maximum discharge and higher flashiness. So it really seemed like predators, and then just the change in that flow regime was what really was causing the changes in our crayfish assemblage. And so there were no longer those high highs and those low lows, and those high highs and low lows is what kind of shapes the habitat as well in these systems. And so if you have just a continuous steady flow regime without any of these changes, then the habitats available and the crayfish and species that are able to survive their changes. So all of that data was on the Bear Creek drainage. So this is data for the Cahaba River drainage. So this is the drainage that was kind of in the more center of Alabama. And our circles represent our unimpounded stream, and our triangles represent our impounded stream. And so in the Bear Creek drainage, we saw this split. We had impounded stream shapes up and unimpounded stream shapes below. But here, as you can see, our circles and our triangles are right on top of each other. So we didn't see that difference in the crayfish assemblage in the Cahaba River drainage. So every time I feel like, do a study, you're telling the story, you do your preliminary analysis, looking at one site, and you're like, yes, this is what I thought was happening. Then you get all the data, and it's like, what is this? And so, yeah, you can tell our nice story of what dams were doing to these crayfish assemblage didn't really play out in our in the Cahaba River drainage. So our question became, so, what's going on in this drainage? Why don't we see these differences in the Cahaba River drainage? And first culprit was we have an invasive species there. So Faxonius virilis is an invasive crayfish species to Alabama, and it was collected throughout both our impounded and our unimpounded stream. And invasive crayfishes can change habitat. They can be using space, food resources, and limited the resources that these other crayfishes need. And so they can really drive the structure of a community. And then this drainage is also right near Birmingham. So there's a lot of development. And so the white here in the map represents the developed areas. And the Cahaba River drainage, 22% of the drainage is developed, whereas only 3% of the Bear Creek drainage is developed. And so development can cause changes to a system similar to how impoundments would change in the flow regime, lots of sediment, going into the stream system, but it can change the habitats drastically. And it's changing these in both the impounded and unimpounded streams. So because both of these streams have other things happening within the stream system, we didn't see any effect of the impoundment. So it looked at it as these outside factors were driving the stream structure, the crayfish structure, more than just simply. So we went on to begin thinking about what is the effects of dams and invasive species on trophic interactions of our native crayfishes. So we have these two factors playing a part in crayfish assemblage. We wanted to know how these native crayfishes were responding, and we decided to look at it on a trophic level. And so we went further into the Cahaba River drainage and sampled a number of impounded and unimpounded streams with and without that invasive species to see how the trophic level of the native crayfish would change. And so trophic ecology is really important to these systems. It gives you an understanding of the functional role of the species in that system, and then the community level interactions. So what does how do these crayfishes impact the food web? What's the flow of energy and nutrients throughout these systems? And so lots of people consider crayfish, basically they're carnivores, they play a lot of different roles, but that all crayfish kind of play the same role is sometimes what's thought. And if you have that way of thinking, then if you lose a species out of a system and you have other species and they're all playing the same role, then if you're looking at an ecosystem function, it won't be a big deal because you still have these other species there. But we plan to, we wanted to look at if these roles would change because of that impoundment and invasive species. And so what we saw was that impoundment did impact crayfish trophic positions. And so we collected 4 different species out of those sites. Canberra's cusa is shown in red, Canberra's striatus is shown in green, Faxonius ericksonianus is shown in blue, and Faxonius virilis, our invasive species, is shown in purple. And so this is a box plot showing the trophic position of species collected in unimpounded and impounded streams, with US being site upstream, DS being sites downstream. And so the higher the trophic position, the higher level they are within the food web, normally the more animal protein that they're eating. And so we did see changes within 2 of our native species, Canberra's cusus and Canberra's striatus. There were changes within their for their trophic position between the impounded and unimpounded stream. And so for Canberra striatus, the green is kind of more straightforward, where basically there was a higher trophic position in our impounded stream. Whereas Canberra's cusa, it was kind of flipped, where our unimpounded downstream and impounded upstream were more similar, and then unimpounded upstream and impounded downstream were more similar. And so, but basically the resources available are changing in those impounded streams due to those lentic areas and the inputs into the systems. For Faxonius ericksonianus, it didn't matter where we collected it, their trophic position stayed the same. But the most interesting thing is our invasive species. It drastically changed between our unimpounded and our impounded stream. And so basically in our impounded stream, they play a much higher role within these systems than in our unimpounded stream. And so they are much higher on the food web and playing a much higher role within these systems. And so our next question was, okay, how is the invasive species impacting our native species? So again, this is a box plot with the colors still representing the same species. And now you're seeing the box plot of the trophic position for native species in sites that did not have our invasive species, and then native species with our invasive species, then our invasive species only. And so natives with and without our red and our green, we didn't see any change if they had, and then if the invasive species were there or not. So it doesn't look like the invasive species is actually impacting the trophic level of our native species. But in turn, our native species is actually impacting the trophic level of our invasive species. And so if we, in sites where we only collected our invasive species, they had a much higher trophic level than at sites where we collected them with natives. And so it looks as if every resource is available to our invasive species, they're gonna play a higher role in the food web. Whereas the role of our native species stays the same. And so if you think about an invasive, the reason that they normally are invasive is because they can survive in different habitats, they can eat lots of different things, and they have this more flexible ability to be flexible in these habitats. And so that's what we're seeing here is that, okay, you know, if natives are here, this crayfish can be fine just eating algae or detrital inputs. But if those native species aren't there eating those things, they will definitely play a much higher role within these food web systems. So what we saw for impoundments are that impoundments fragmented the genetic populations, that they homogenize our crayfish assemblages, and they cause shifts in the trophic position of our native crayfish species. So with the data that I collected from this impoundment study, we had a unique chance here to look at our species genetic diversity correlation. So we had species genetic population data, we had data on richness and abundance of the species within these systems. And so we were able to tell if we have some type of correlation between the number of species, how diverse the genetic populations are, or if the abundance correlated with genetic population, or if the number of species correlated with abundance. And so if you find correlations between these different metrics, for someone who's a conservationist, someone who's a manager, it may be that, oh, if I know if I have a lot of species, I have a high genetic diversity, I can just go and sample a stream and say, yes, we have high species diversity. Then these other metrics that are important to crayfish conservation are also doing well. And so we wanted to see if there were any correlations so that we can inform if you need to be sampling all of these different, like taking all these different metrics, or if one could kind of give you an idea of the others. And so, so the focus of management is normally richness and abundance. So you go out in a stream, you do your surveys, and you know how many species are there and the abundance of those species. Most people don't have the funds and the expertise to go and dig into the genetic diversity of a population. And so if these things correlate, it could give managers an idea of the genetic diversity of a population. If they don't correlate, it may mean that, okay, we need more genetic studies, because if the genetic diversity is very low, even if you have a high abundance at the time that we sample, we still could see drastic changes within these populations. So here we use the Chow-1 rarefied richness for our species indicator, and this indicator estimates the number of species based on your collection. So if you have a lot of rare species, it'll estimate those that you may have missed within your sampling. Then we use haplotype diversity, nucleotide diversity, and polymorphism as our genetic for our genetic data, and then crayfish density was our abundance measure. And so these are from data collected in through the impoundment study, and our genetic data came from 2 species, Faxonius ericksonianus and Faxonius vallis. And they were the most abundant species throughout those temperatures. So these are the correlations, the Pearson R is all of the correlations for the different genetic, the correlation metrics, and then p-values are on the right-hand side. So basically, most things were not correlated at all. And so I want to point out here, two, that there was one that was significant and one that was, you know, almost there, but there was a negative correlation between the abundance of crayfish we were collecting at a site and the genetic diversity at a site. For Faxonius vallis, just for one of the species we assess. And so the higher the abundance of all the crayfish we collected, the higher abundant they were, the less diverse the genetic population of the species were. So it looks to be that, oh, just because you have an abundance of crayfish does not necessarily mean for the crayfish species you're assessing, that the genetic population is the same thing. So, but mainly we didn't see any correlations. And that's really a bummer for conservationists, because that means, okay, we need to be doing genetic assessments. We need to be understanding the genetic diversity of these populations. And, you know, has there been a bottleneck, or are there things impacting genetic diversity that's not impacting abundance or species richness? And so with this assessment, there were crayfish in impounded and unimpounded streams. And so there could be some type of impact or change to these correlations because crayfish were somewhere in impounded and unimpounded streams. We couldn't assess them separately because of our sample size, so we assessed them together. But we went back and looked at environmental factors that could be driving species abundance, richness, and genetic diversity to see if that differed between impounded and unimpounded streams. And so these are graphs that show the factors that did differ between impounded and unimpounded streams for each of our different metrics. And so the first graph shows the child one species richness and stream width. And so the gray is our unimpounded stream, and then the white is our impounded stream. And so what we see here is that stream width, the larger the stream width, the more species that were collected in our unimpounded stream. But the larger the stream width, the less species that were collected in our impounded stream. And so, you know, the larger, because of these differences, could play a part in why we didn't see differences between our between our correlations, between species richness and other correlations. And so our next graph shows the abundance of species. And so we see here that the more large woody debris we had in our unimpounded stream, the less abundant crayfish were, but there was no correlation in our impounded stream. And then for medium substrate size, the larger the substrate, the more crayfish collected in our unimpounded stream, but no correlation within the impounded stream. And so basically what this shows is that there are different factors impacting the populations within impounded and unimpounded streams. So it could be that we just need to go back and do this assessment separately for each stream type. So it could be that there are some correlations in our impounded stream or in our unimpounded stream, but they're kind of broken up in our impounded stream because other factors are impacting this crayfish population. And genetic diversity was actually the only diversity metric that was impacted the same in the impounded and unimpounded stream. So how does this impact conservation? Stream crayfish impact diversity metrics, stream characteristics, impact diversity metrics. So depending on the characteristics, stream width, large woody debris, the diversity metric respond to that differently. And fragmentation impacts these relationships. All species are not impacted the same by environmental drivers. We saw that negative relationship for Faxonius validus, but we didn't see any relationship for Faxonius ericksonianus. So you can't just do a genetic analysis for one species, you really need to understand all the different species within the community. And then there was high species richness. Just because you have high species richness or abundance does not mean that that's correlated to high genetic diversity. So you can't just assume that a population is doing well because you assess one of those measures. There could even be negative relationships in some cases, and genetic diversity, it must be assessed by conservation to conserve conservation. So we need to increase those types of studies to truly understand how these populations are responding. And so my last thing I want to touch on is a new project that I am starting with Brandon, Dr. Peoples. And so we just got funding for this project. There's no data. But we'll have a few graduate students joining the department next year or in January. And so I want to give you a little overview of what they'll be working on for this study. And so we'll be assessing the use of environmental DNA as an early detection method for aquatic invasive species. And so here is a map of the southeast US with the green represented national forests. And so national forests, they play a big part in the refuge for aquatic biodiversity with high species diversity and endemism found within our national forests. So these forests provide critical habitat for a lot of threatened and endangered species. But when you think about the movement of aquatic invasive species, a lot of times it's correlated with people. And so the National Forest also provides lots of recreational areas for people. And so these might be our highest risk locations where people are fishing, boating, camping, water holes, where the movement of these species could be happening. And so currently there's no aquatic invasive species monitoring protocol for the National Forest within our southeastern region. So the goals of this study is to evaluate the effectiveness of eDNA to detect aquatic invasive species in wadable streams. And we'll be using water samples and then seeing what we see through DNA in those water samples. And then we'll be assessing the detection of using multiple aquatic invasive species, so not just simply crayfish, but fish and mollusks as well. And then we'll be comparing those to our traditional sampling, electrofishing, quadrat snorkeling surveys for mollusks. So do we need to, DNA would be quick and easy, you can just take a water sample, but do we need to really be out there doing these traditional methods to detect these species or not? Then we'll develop occupancy models and hopefully identify these high-risk areas for invasive species introduction. And then through that information, we'll develop a protocol that could be used throughout the southeastern US. So who will use this? It'll be, it's geared for the Forest Service, but we're hoping that other federal and state agencies and private landowners will be able to use this tool. And then we will disseminate data through workshops, webinars, field tours, presentations, and publications. And so with that, my key points from all of that was that crayfish play a very important role within our aquatic ecosystems. And losing them from a system can change the system's food web structure and the overall function of the system. Land use and water management can fragment these populations, so we need to think about ways to keep populations connected. And then impoundments impact crayfish genetic structure, their assemblage, and their trophic position. So the degree of impact depends on anthropogenic impacts, species dispersal tendency and habitat preferences, and the size and age of impoundments. But when managing, we need to mitigate fragmentation and mimic our natural historic conditions to promote crayfish population growth and dispersal. And with that, I'd like to thank our Forest Service team, the South Carolina National Forest, many university partners, and others. I'm taking questions.
So you kind of came back at the end of your talk, to something I was thinking about early on, which is the role of people moving crayfish around, right? So we know people use them as bait, and I was wondering if you gave some thought to kind of the population genetics in that first study where you're seeing in one kind of system, looks like they're more connected than another, and what role might people play in those patterns?
So so people play a huge role for that species. I really don't think people play much of a role with moving them because they're a burn species and they are hard to collect. So you gotta really, I mean, I've seen people dig down a meter to get you have to really do a lot of work to collect those species. And so I don't think there's a huge role with people moving them around. Now our stream species and our species in vernal pools, that could be a huge deal. It's a lot of the reason why we think we have Vaxonia spiralis, that invasive species in Alabama, because that's a big species, and our bait shops aren't thinking about where they got the species from. You know, people just going getting bait, and then I'm done fishing and I still have bait. I throw the rest of it in the stream. So we do think that's a big reason that we're seeing a lot of invasive species, the places we're seeing them. And then also teachers and science teachers, after you have your crayfish, are easy thing to have as a class pet. And then what do we do with that in the year? So we've been working on a lot of education outreach to folks to understand that, okay, you know, once you're finished fishing, once you're finished with this species, it's best to, you know, go get some ethanol and and not just throw it back out in the stream unless that's where you caught it. And so, you know, a lot of these crayfishes they may be just made up to a certain drainage. So just going 20 miles down the road may be a big difference. So it's not even that, oh, I'm taking something from, you know, Mississippi and bringing it to South Carolina. It could be right, you know, right in your backyard that you think is really close. So I got it there, I'm just dumping it there. But it could be a big issue to those systems. Yes.
So you saw with the invasive species, how whenever it was just it in the stream, it had a really high trophic level. Do you think that's gonna be a problem if you ever try to do like reintroduction? Like, would the reintroduced species be able to bring the trophic level of that species back down, or would it just devour all the food and start the reintroduced species out again? Or if it
That's a
happened
good question. So with not having any like data on any types of reintroductions that have been done for the crayfishes, my thought is that because they're so flexible, they would kind of just drop down to what's available. But I think it would probably play a big part on how abundant that invasive species is, if you know, if the natives are able to compete well with it or not. And so yeah, I'm not sure if they would shift. I would hope that they likely would. But yeah, that's a good question.
Kind of following up on that last question, the streams that only had the one invasive species, were there obvious differences within stream habitat, watershed land use in those systems that seem to correlate with that, or?
No, and I should say there's we're working with Troy University, so they've been out at most of those sites. I hadn't been to all of those sites. But from talking to them, there aren't any, they haven't seen any like just obvious differences in those environments. And you know, honestly, we're kind of surprised they were only collecting the invasive species there. And so we don't have data on if there used to be native species there or not, to know if they just outcompeted them and now they're the only species there, or there wasn't anything else there originally. So yeah, they hadn't talked about any just drastic differences where they thought they, you know, why.
See, so there's 90 species of crayfish in review for ESA listing. That's crazy. Do we have many in South Carolina?
There are, Yeah, there are a few in South Carolina. There are 3 disco Canberra species, and then a few others that you find like in, not far from here, actually, kind of like going up into the mountain area that are listed. So I would say, I think maybe like 7 or 8 would be in South Carolina.
All right,
Thank you.
thank you once again.
It's bigger. Yeah.